This is lecture 13 entitled Infectious Disease. We could have done this one earlier in the semester when we were talking about viruses and viral diseases as well as fungal diseases, but many of these topics are going to be applicable more to bacteria, and so that's kind of why we're doing it now. We're transitioning. After this lecture, we move on to bacteria and the diseases that they can cause. But some of this information equally applies to fungal infections and also viral infections. So it's actually kind of a nice to have in the middle, so it kind of straddles all of those things. One thing you may not have thought about, or maybe you have, is we have millions, billions, trillions of microorganisms living on and in our body all the time. And this is something that we have talked about previously that's called the microbiota or the microbiome. And so there's really just a small fraction of microorganisms that can actually cause disease within humans. And so one way of looking at this then is thinking about and asking the question, what is it about those microbes that allow them to cause disease in humans when there's so many others that live on and in us that don't cause us any harm at all? And so that's the ultimate goal, is to understand what about certain microbes allow them to cause disease. And so before we do that, we want to make sure we understand these definitions. So I've used the term disease. And so that's going to be some clinical manifestation, some alteration in our physiology that you can perceive you're not feeling exactly right. So we've all experienced this, whether it's been a gastrointestinal issue or a respiratory infection, or even something with your skin. You know when something isn't going right there. And so that's because there's been some change in your physiology that you can then perceive that is a disease state. Oftentimes that disease is going to occur because there's been an infection. So an infection is just described when a microorganism, more specifically we call them a pathogen, so a microbe that we know can cause disease, is successful in infiltrating us or penetrating those host defenses. So it's entered our body by some mechanism. And that's what we're defining as an infection. So just because you have been infected by a particular pathogen does not mean you're going to get the disease or not. There's a variety of things that can influence that. As you might guess, it's dependent upon how many of those pathogens have actually entered your body. So the more that you're exposed to, that could contribute to causing disease. How good is your immune system? So do you have some protection? Have you been vaccinated? What's your overall immune status if you're immune compromised, etc. So those are some terms we're going to be using. Going back to the microbiome, we've talked about how we define this. These are that group of microorganisms that are found on and in our body. And again, this is an important part. They don't normally cause disease. There are instances where members of the microbiome can cause disease. We saw examples in the fungus, where we talked about candida, which is a yeast that grows on different parts of our body, can cause disease if given the right opportunity. And so these microbiome members play an important role. So they basically are taking up space so that other microbes, especially those harmful ones, can't actually get a foothold and establish themselves there. So even if they do successfully invade, they may not be able to grow very robustly because of all of these other microbes that are present that are kind of taking up space. And so that's this microbial antagonism that we're talking about. So it's kind of competition for space within our body. In terms of the microbiome, we've seen this before, and I just want to remind you of where it's located. So this is a list of where we know the microbiome members exist. And so it's really anything that has connections to the outside environment. Outside environment. And so obviously the skin being outwardly facing, and all those mucous membranes, so whether that's the oral or the nasal cavity, the respiratory tract, the gastrointestinal tract, parts of the reproductive and the urinary tract, etc. And when we talk about microbiome, it really encompasses all of the different microbiology groups. And so we tend to focus mostly on bacteria, but there's fungal species, there's protozoans that are present, and there are definitely viruses. And so while this is a list of locations within the human body where we know there are microbiome members, these are going to be areas that we presume are free of microorganisms or sterile. So the heart and the blood and the muscles. And so as you might guess, these are locations that are not, well, most of these are not directly connected to the outside environment. And so they are going to be free from, excuse me, microbiome members. We've talked about this before, but just to remind us then, so in the womb, so as you're developing inside your mother's womb, it's generally free of microorganisms. There are some infections that can be transmitted across the placenta, but what we're talking about is kind of normal development. It's going to be free from microorganisms. And so that means that during the birthing process is when the colonization of microorganisms to become microbiome members is going to begin. And so whether that is during passing through the birth canal or being delivered through a cesarean section, as you might guess, that's going to lead to a different exposure. Breastfed versus bottle fed could also lead to different microbiome members. But all of those new situations and new environments are going to lead to the colonization of microbiome members. There's a great deal of work that is looking at how early establishment of the microbiome leads to long-term consequences in terms of our health. And so whether that is early in life and establishment of those microbiome members in the respiratory tract and their role in developing respiratory issues later in life, same with gastrointestinal issues, etc., maybe even skin issues. And so we don't know all of the answers to that, but it's a really interesting possibility of how these microbiome members play a role in the normal physiology of many of these locations. And so that's something that's going to be becoming more and more important in the future for sure as we learn more about these interactions. So we've talked about some of these definitions, but we want to add to it. So we really want to differentiate those microorganisms that are just part of the microbiome versus those that can cause disease. So we've used this term before. A pathogen describes a microorganism that can cause an infection and can cause disease. So they have something about them, some characteristic that enables them that if they do get into our body, it doesn't guarantee they can cause disease, but they potentially can. And so we can talk about then this characteristic, so it's called pathogenicity. And so this is kind of a yes or no question. So you can ask, does a given organism exhibit pathogenicity? And so it's just another way of saying, is it a pathogen or not? So can it cause disease, yes or no? But then to dial that in a little bit more, we use the term virulence to talk about that relative scale. So how likely is it to cause disease? How virulent is it? And so just because something's a pathogen doesn't mean everything is equally virulent. And so the term virulence is something we use to describe that spectrum of how likely is it to cause disease if it does enter our body. And when we were introduced to fungal diseases, we saw this term. And so those terms aren't only related to fungal infections, but we talk about true pathogens versus opportunistic pathogens. And so the same definition applies to these opportunistic pathogens. They are going to need some special conditions, and they are described down here. And so, again, it all relates to some immune dysfunction for the most part. So the immune system isn't working, or there's a disruption in the normal microbiota, and so that space that we were talking about, the competition for space, is disrupted, and so new microbes, potentially pathogenic ones, can get to that space and colonize and then cause a disease. Or those microbiome members are transplanted to a location that they are not normally found. And so any of those or any combination of those can lead to an opportunistic pathogen becoming the cause of a disease. So when we talk about an infection, there are several steps involved. That's kind of a circle of infection. So if you think about how a disease can be transmitted from one person to another, so to speak, there are going to be these steps. And there are 5 that are listed here. The first step, obviously, is there needs to be some mechanism of entering. And so we're going to talk about how diseases can be transmitted. And so depending upon the disease, there are different ways that that can happen. But most of you are familiar with different routes of entry. So some you can get infections in your skin, or it can be foodborne or waterborne. So it's going to target the gastrointestinal tract, or obviously with corona, respiratory tract infections, etc. So there needs to be some mechanism of entry. Once something gets in, it's really helpful if a microorganism has some characteristic that allows it to attach. And so if it can attach, that's going to be really important in establishing an infection and then potentially progressing to a disease. So for example, if it's a gastrointestinal disease, can it actually physically bind to those epithelial cells along your gastrointestinal tract? If it's a respiratory infection, can it bind to those respiratory epithelial cells along your lungs and breathing airways? And that can be said for any potential target if it's a urinary tract infection as well. Once it's there and attached, then we do have an amazing immune system that helps prevent infections becoming diseases. And so many microorganisms have found ways of avoiding or evading the host immune response. And so if microorganisms contain characteristics that allow them to do this, then as you might guess, those microorganisms are going to be better able and better situated to cause disease. The next is actually generating some substances that lead to a physiological change. So as we defined, that disease is going to be something that you can perceive and feel. So whether that is generating a fever, or whether that is getting a rash, whatever it might be, ulcers, blisters, a cough, you guys all have experienced different signs and symptoms of disease. So often it's related to chemical compounds that are produced by specific microorganisms that lead to this disease progression. So that's what we mean here, direct damage. So this is coming from the microorganism, but there's another really important category that we classify as indirect damage. And this is the result of immune chemicals that are being made to fight a specific infection. And so this is changes that we see in our physiology that are indirectly due to the presence of the microbe. Not that the microbe is causing the change itself, but it's the presence of the microbe causing an immune response, and that immune response is then what you perceive as signs and symptoms of the disease. So for example, fever could be one, or inflammation another, etc. And then the last bit of this, for a microorganism to be successful in causing disease and continuing to spread, it needs to be able to leave a host. And so this is just called portals of exit. And there's different ways that microorganisms can leave the human host. So again, this tends to be disease and microbe specific, but most people are aware of the ways in which microbes could potentially be spread. Another way of looking at this is this term called virulence factors. So remember, we just defined that term virulence, which is how likely is something to cause disease. So how virulent is it? And so a basic way of answering that question is, well, how many virulence factors does a given microorganism have? And so virulence factors are going to be characteristics or traits that are exhibited by pathogenic organisms that make them better able to establish an infection and then potentially progress to a disease. And so they're going to be very much related to these different categories. So do they have something that allow them to attach? And so if they do, that would be one category of virulence factors. So we've talked about things like fimbriae or capsules or spikes, etc. Another category of virulence factors, we can talk about the ability to avoid phagocytosis or other important host immune responses, so those are virulence factors, etc., or the ability to make toxins or enzymes that allow them to survive. So we're going to talk about different categories of virulence factors and how they kind of interplay with this progression of infection and disease. But these virulence factors are going to be most important in these middle three. As you can see, this is where it's really allowing a microbe to establish an infection and potentially cause disease. And so the more of these characteristics or virulence factors that a given microbe has, the more likely a microbe or pathogen is going to be able to progress between these consecutive phases of establishing an infection and disease. So these are different categories of virulence factors. And so over the course of the next several slides, we're going to break down each of these into more specific examples. And so as you might guess, being able to attach or adhere is a really important virulence factor. That was, again, this step right here, attaching firmly. And so adhesion factors are going to be one category of virulence factor. They're kind of related to biofilm, but we break them into distinct categories because as we have seen before, biofilms allow microbes to attach to one another and form this sticky web that can form a really complex community of microorganisms. So it may not be just a single microbe, but rather a collection of interacting microorganisms that leads to an infection and potentially disease that's more than just the individual parts, but they're working together in a way that makes it harder for our immune system to get rid of them. It's harder for antibiotics to work against them. And so we know biofilms can be a really important part, or forming biofilms is an important part of causing disease. So this is the only one that I don't have an extra slide for. And so that's about all I'm asking you to know, that it's a category of virulence factor and that basically what a biofilm is and that it can allow bacteria to cause disease. But these other ones, so adhesion factors, we have a couple slides on, and then we'll move on to anti-phagocytic factors, so we'll talk about that, and then enzymes, and then toxins. So all of these other ones have additional slides, so we'll get a little bit more detail. These pictures over here are going to be reinforced on additional slides as well, but as you can start to see, these are different examples of these virulence factors that can lead to disease. So here's a bacterium. So these little, the larger green circles are bacteria that have invaded. So say this is your gut. So it's something that you ingested. So contaminated food. So what this is showing is the bacteria are making an enzyme that can actually digest the glue holding these cells together. So that cement or glue is normally going to prevent microorganisms from gaining access to deeper levels of tissue in your gastrointestinal tract. But if a bacteria makes an enzyme that dissolves that, then as you might guess, you can get invasion, and that can lead to a progression of infection and potentially a disease. So rather than talk about each of these individually on this slide, we'll see them in future slides. So the first is going to be adhesion factors, but what I'm going to do is talk about these portals of entry first, and then we'll get to attachment and adhesion factors there. So portals of entry, you likely know these, whether you've categorized them or not, but we are going to be exposed to microorganisms all the time. So when you can ingest food or drink water, or any other fluid, it's not sterile. There are microbes in it. How many microbes and what the composition of those microbes are going to vary. The same is true for any air that you breathe or any cut that you get. There are going to be microorganisms that are able then to infiltrate your body. So the most common routes of exposure then are going to be the skin, which is a really important physical and chemical barrier that prevents many microbes from actually getting to certain parts of our body, so it does a great job. But obviously, you've experienced this. You get nicks and abrasions and different injuries that you sustain to your skin. And so once there has been that physical disturbance to your skin, it allows a specific site of colonization potentially. most microorganisms can't just penetrate skin on their own, but we did see several of these that can. If you think back to the parasites, hookworm was one example. That was the one that could burrow through the feet of barefoot individuals as they're walking through soil that has the larvae in it. Those larvae can burrow through the bare skin and then establish an infection, ultimately in the intestine of that infected individual. We also saw the blood fluke, or schistosoma is the genus of it. That one, while you're swimming, that Cercariae, it had a shape that looked something like this. It's kind of got a triangular head and then a forked tail. So that was the Cercariae of Schistosoma manzoni, and that could burrow through the skin if you are in contact with infected water. So not all microbes can penetrate intact skin, but as I mentioned, there are plenty of instances where we get cracks or cuts where it could lead to an infection. So that's the skin. Obviously, I'm sure we've all suffered from gastrointestinal, sometimes it's called stomach flu, but it's gastroenteritis of some sort. So that can be viral, it can be bacterial. And these are just microorganisms that we have ingested through food or beverages. Obviously, the gastrointestinal tract has a certain order that things are going to pass through, and the stomach is pretty early on. So those pH changes, again, the low pH of the stomach, that acidity is not hospitable for all microorganisms. So that serves as a helpful barrier and chemical prevention. We have digestive enzymes, but there are plenty of microorganisms that are adapted to survive in that. So plenty of diseases that have plagued human society throughout the ages are passed through food and water. And then obviously the respiratory tract is one that is going to be probably the most commonly transferred, or the most common types of infections. So respiratory infections, cold and flu, have been around for many, many millennia, and we all have experienced them. And we obviously have evolving and re-emerging microorganisms, or newly emerging microorganisms, like these novel coronaviruses that we're seeing more recently, that are also respiratory infections as well. So these can be transmitted from exposure to the oral cavity, the nasal cavity, the obviously upper respiratory, the lower respiratory tract as well. So those are going to be the most common, and we've probably experienced each and all of those. There's obviously sexually transmitted diseases as well. And then I mentioned that the womb is relatively free of microorganisms, but there are some that can be transmitted across the placenta, so it's called transplacenta. And then also during the birthing process. So if mom has certain infections present, then they can be transmitted during that birthing process as well. So the last thing here is talking about infectious dose. And so this varies depending upon the microbe that we're talking about, but infectious dose is just defined as the minimum number of microorganisms necessary to establish an infection. So again, some don't require much at all, so it's very infectious. So a low number leads to an infection, and then that can lead to disease. But others actually require a higher dose. So a low dose isn't going to lead to an infection or disease. So now we get to that second step, the ability to attach or bind. And this is going to be very much driven by one category of virulence factor, as we mentioned. But the ability to bind to either one another, so bacteria binding to bacteria, so sticking to one another, or, I guess it can be and as well, and/or, binding to host tissue. And so this is driven by a specific interaction between a molecule on the surface of the microorganism and a molecule on the surface of host cells and tissues. So this is very much the analogy that's often used is a lock and a key. So you have a molecule on the microorganism that fits perfectly into a molecule on the host tissue. And so once this has occurred, that is going to then allow the microorganism to establish an infection and maybe just remain localized there. But if it establishes an infection in one area, it also leads the possibility that it could spread beyond that. It could invade to other tissues. And so being able to attach is really important. So these are some examples that we've seen in the past. So fimbriae are those, well, they're numerous, short hairs for attachment. So not all bacteria have them, but those bacteria that do have those fimbriae, and you can see in this picture, fimbriae are for attachment. So if a bacteria has it, it's going to allow them to attach to one another or to host tissues potentially. We learned that capsules, I'm going to expand this a little bit, or slime layers, these were our two categories of glycocalyx. And so both of them had sticky properties to them. Slime layers, as you might guess, are a little bit more sticky. Capsules are less sticky, but still allow for attachment. So if bacteria had a slime layer or a capsule, it could allow them to attach. So here we have a host cell and those bacteria are attaching. But these adhesion factors are not exclusively for bacteria. So we talked about viruses having unique molecules on their surface known as viral glycoproteins or viral spikes. And so here is an example of a virus that has these viral spikes. This looks like an enveloped virus. And that enveloped virus has those viral spikes on it, and this would be a polyhedral capsid with some nucleic acid on the inside. But those viral spikes are really important in binding to and attaching to the host tissue. This is not to memorize, but just to show you, most people recognize some of these diseases. So gonorrhea is a sexually transmitted disease. Diarrhea and dysentery are both gastrointestinal diseases, pneumonia, etc. But these are the causative agents, so the microorganisms that cause the disease. And many of them have some mechanisms of attaching. And so that's really critical for their ability to establish an infection and then progress to a disease. So these things, these virulence factors, we can categorize them as, this is a category of virulence factor. So they're called adhesion factors. And they can be specialized structures like we talked about, fimbriae or glycocalyx. Those glycocalyx, we have two examples, slime layer or capsules. But it may not be organized into an entire structure. That can be a possibility in some microorganisms. It may just be specialized proteins that are present, like in the case of viruses. We said they had those viral spikes, but bacteria can have them too, and they get a special term. So attachment proteins in bacteria are called adhesins. So that's a term that you want to learn, and it's kind of, in that word, it looks like adhesive, just like we have for adhesive tape. So it hopefully makes sense that adhesins are going to be, they're going to function in attaching. So now we just have to layer on top of that these are proteins on the surface of bacteria that allow that to happen. And so this is kind of showing how this could potentially interact. Here's a microorganism that have some molecules on the surface. And the generic term we use in biology is ligand and receptor. And so this is, again, like a key and a lock. And so ligand receptor is used generically across biology to talk about a really specific interaction between two molecules. Another example of, just to connect this to other courses that you might have, ligand receptor. So in this case, what we have is something like an adhesin on the bacteria to some protein on the surface of a cell. But you can also talk about, in normal physiology, you can talk about a hormone like insulin interacting with its own receptor, so insulin receptor on the surface of cells or something like that. So these terms that are being used here are not specific to microbiology, but it's much more broad than that. It is describing an interaction between two molecules. And so ligands and receptors. And so it is that specificity between ligand and receptor that can help explain why a particular microbe can lead to a particular infection and then a disease. So obviously having the right keys for a respiratory tract is going to be really critical for a respiratory infection. Having the right keys for a urinary tract are going to be pretty important for urinary tract infection, etc. So that helps explain why certain microbes can only cause certain, can only enter our body a certain way to be successful, and then why they lead to a certain clinical progression of a disease. These last two just point to the importance of these adhesion factors. So if we change or we block the ability of that bacteria or microorganism ligand, so that key, if we block the key, it can prevent the process of infection and disease. So the analogy would be if you stuck gum or you took the key and kind of filed it down, that key no longer works. And so it no longer can lead to the progression that we described before of binding, establishing infection, and potentially causing disease. So an extension of that, the inability to make those proteins, attachment proteins or adhesins, is going to take an organism that was virulent, and now it can turn it into what we call avirulent, and that A means without or lacking, so lacking virulence. So these categories of virulence factors that are called adhesion factors are really critical in establishing an infection and then progressing to a disease state. So hopefully I've made that point pretty clear. So that's one category of virulence factors. But then once something does start to establish an infection, if it can attach or being able to survive, different immune processes is also going to be really important in establishing infection and progressing to disease. And so without going into a lot of immunology right now, we're going to see this more towards the end of the semester. This is a generic term we use to describe some immune cells. They're called phagocytes. And they have the ability to carry out this process called phagocytosis. So they're phagocytic cells. And for the time being, all you have to know is these are white blood cells. And they go around and they eat and destroy. And so I usually use these two: ingest and digest pathogens. So that's what these cells are trying to do, these phagocytic white blood cells. They ingest, and that just means the microorganism is actually internalized into the cell, so it's brought into the cell, so the cell has to engulf it or eat it. So I mean by ingest. And then once inside, that phagocytic cell is going to dump a bunch of chemicals that are going to normally digest and destroy the pathogen. So that's what we mean by ingest and digest. So that's how it's supposed to work. But different microorganisms have found ways of disrupting this process to avoid this really critical immune function. And so these are then classified as anti-phagocytic factors. And this too is another category of virulence factor. And so these virulence factors are going to be used by different pathogenic organisms to avoid this process of phagocytosis and avoid the host immune response. So one we've actually talked about before. So we have said bacterial capsules are going to be really important. So this was the tightly bound glycocalyx. So we contrast that with the slime layer that's more loosely attached. And we also said capsules allow bacteria to avoid the immune response. So that's kind of what we said at the time when we learned about capsules, but now we want to get a little more specific about how that can be achieved. So here are two examples of how different capsules could actually achieve that outcome of avoiding the immune response. So one is if a bacteria makes a capsule out of chemicals and molecules that are normally found in the body, then it's going to be much harder for our phagocytic immune cells to actually see that particular bacterium as foreign and therefore less likely to attack it. So if you haven't thought about what immune cells have to do on a daily basis, they have to make a decision and decide, do we attack this cell or do we not attack this cell? And the goal is to not attack normal, healthy human cells and leave them alone, but it has to identify bacterial cells, especially pathogens, as being foreign and actually attack them and destroy them. And so if a bacteria is masking itself, cloaking it in molecules to make it look more human-like, that's a much harder process to do. So that's what some bacteria do, and that's how their capsule works. But another is shown in this picture. And so as we described, what a phagocytic cell has to do is it has to find a bacterium and then physically ingest it and actually move it in. And so some bacterial capsules are much harder to physically adhere to than bacteria lacking capsules. And so if a capsule is made up of molecules that make it, as it says here, more slippery on a molecular level, then it's harder for the bacteria, for our immune system to ingest and digest. So think about what we've talked about so far. The goal is to ingest and digest. So it's a game of hide and seek for starters. And so some bacteria are better able to hide from the immune system because they have these chemicals that don't look foreign. They look like they're supposed to be there. But other bacteria are easily, I don't say easily, but more easily recognized as foreign, but they're just harder to grab onto and actually internalize and ingest. So that's what's being shown here. So in either of those two situations, that capsule gives a really important advantage to the bacteria in avoiding the immune system. So that's how some capsules work. There are also chemicals that serve a role as well. So these are chemicals that can be secreted by the bacteria that leads to having antiphagocytic effects. So here's, again, two examples. So here's an A and here's a B. So some chemicals can actually prevent, I'm going to read this to you and then explain it, the fusion of the lysosome to phagocytic vesicles. So what the heck does that mean? Well, this is the digest part of the ingest and digest. So this part focused on bacteria's ability to avoid the ingest. Some bacteria actually are ingested just fine. So here's a bacterium that has just been ingested. But now it's going to disrupt that digestion part. And so this bacteria can release chemicals that prevents the fusion of this organelle. The lysosome contains digestive enzymes. So it's that lysosome that's going to play a really critical role of the digestion part. But if the bacteria prevents that lysosome from actually fusing to this and dumping the chemicals on it, so normally this lysosome would come in here and dump all of its chemicals like so. And that's an important part of phagocytosis. But what we're saying is some bacteria make chemicals to prevent that from happening. So they actually not only survive, but they can potentially reproduce inside of our immune cells in kind of a protected, enclosed cellular environment. So that's one example. This one is a little more severe. So leukocidins, if hopefully you notice that ending right there, we've seen before. So when you see that ending side or cidal, you should start thinking of death. And so leuko relates to leukocytes or white blood cells. So if you haven't remembered those terms, leukocytes equals white blood cells, which are your immune cells. Oops. And so leukocytins are going to be chemicals produced and released by specific microorganisms that are going to kill white blood cells. And so since white blood cells carry out this process of phagocytosis, it's a much more extreme way of avoiding an immune response by killing those cells responsible for carrying it out. So anti-phagocytic factors also a really important virulence factor and a part of establishing an infection. Now we're moving on to enzymes. So we talked about enzymes some time ago. For exam 1, I had you review some enzymes, but remember, enzymes allow some reaction to be carried out at a faster speed. And so these enzymes, we call them exoenzymes or extracellular enzymes. And all that means, when you see this prefix, it means outside, and you can almost see the word exit in it. So exoenzyme is going to function outside the cell, or another way of saying that is they have to exit the cell prior to them having their function. So some bacteria make exoenzymes or extracellular enzymes that can have important consequences in establishing infection or progressing to a disease state. So these are, again, two categories that we are going to see examples for. So if an enzyme is going to dissolve and destroy some structural chemicals in the body, that's going to be pretty important because it may allow them to invade to deeper tissues. So that's going to be an important consequence. Or enzymes may allow for avoiding defenses again. So this is where the categories overlap a little bit. That sounds a lot like the previous category we just talked about, anti-phagocytic factor, and indeed it is. But we also want to talk about it here because specifically it's an enzyme that bacteria are making. So we'll give you some examples of each. So here are 4 examples that we're going to talk about, and they're really paired. So the first 2 we can kind of learn together, and the next 2 we can learn together. But do notice all of them have that naming scheme that we learned earlier. If it ends in ASE, we're talking about an enzyme. Okay. And so the first example is hyaluronidase and collagenase. So these are both bacterial enzymes, or we can be more specific, exoenzymes. And you may notice how each of these start. Collagen and hyaluronic acid are both molecules that hold cells / tissues together. So collagen is a really important protein that helps bind our cells together. It's from your anatomy and physiology class. It's something we call the extracellular matrix. And so cells link together to form tissues, and tissues link together to form organs. And so we need these cement or glue-like molecules to help hold everything together. So collagen is one of those important molecules. And then so too is something called hyaluronic acid. So each of these is an important component of the glue, if you want to use that term. Okay. So now that we go back to the bacteria, hyaluronidase is a bacterial enzyme that can digest the glue component hyaluronic acid. Collagenase is a bacterial enzyme that can digest that glue component called collagen. So here's an example of a bacterium that has entered our body. And so this is just generic, but you could think of it as entering our respiratory tract. So this could be the airway passage. It could be the digestive tract. So this would be then the lumen of the gut. It could be the the skin, the surface of our skin, whatever the case might be. So just a generic bacterium that makes this enzyme, hyaluronidase. And so normally there's this glue that holds this cell and holds it down into that basement membrane. And so if a bacterium can make that hyaluronidase, then it can digest that glue holding these cells together. And so the bacteria have the ability to get deeper into the tissue than they would if they didn't have that enzyme. And then if you have that same bacteria also make collagenase, well, if collagen is important in holding these cells together down here and this tissue together, then now the bacteria can invade even deeper. So not every bacteria makes hyaluronidase. Some make only hyaluronidase, some make collagenase, some make both, some make neither. So again, you can think of this as a profile of a specific microbe, and you can ask, which virulence factors does it have, does it exhibit? And the more virulence factors a particular microbe has, as you might guess, the more virulent it is, the more better able it is to cause disease. And it also helps explain how the disease can kind of clinically manifest itself. Is the microorganism capable of spreading beyond the initial site of infection or not? So these two enzymes, hyaluronidase and collagenase, are going to be important in playing this role of dissolving structural chemicals or glue within our body. These next two are going to be important in hiding from the immune system. And so coagulase is an enzyme. And if you look at this prefix, it also gives you some clue. It has the word coagulate or the same root. So coagulate is the same thing as clotting. And so coagulase is going to stimulate that process of forming blood clots. And so if there's a bacterium present, here it is, that makes coagulase, then those clotting proteins are going to start to come out of solution and form a clot around this cluster of bacteria. And clots are something that are part of the normal physiological process. And so while there is a process to remove clots over time, they're not removed immediately, but it takes some time. And so that gives some time and some protection where the bacteria can grow and divide basically undisturbed from the immune system. And so the bacteria can increase in number over time, basically, again, hiding from the immune system. So this is an enzyme, an exoenzyme, that allows for an anti-phagocytic factor effect because it allows the bacteria to hide. Many bacteria that make coagulase also make the enzyme kinase. So at a later date, they can make and release this enzyme that will cause them to digest the clot. And so now what you have is an increased number of bacteria that could potentially invade into other areas because they now have a lot more bacteria present and it's harder. Just think in terms of pure number in armies. If you have a large army, that goes a long way in helping win a war, or at least winning a battle of a war. And so that infusion of an increased number of bacteria can be an important part of explaining why you might get a clinical manifestation. So these are all enzymes, as evidenced by their ASE ending, and they have different effects. Some allow the bacteria to invade, some allow the bacteria to potentially hide. Not all bacteria have each of these, but now you can start to see the list of the more of these virulence factors that a bacteria contains, the more virulent it might be. So we have one more category, which is toxins. And just to differentiate what we said, so toxins are going to be some chemical product of a bacterium or of a microbe that's going to lead to damage or harm of host tissues and host cells. And there are two types of toxins: an exotoxin and endotoxin. So notice these are really similar to one another, and so you really want to make sure you learn the difference between them. But again, we see this prefix that's going to differentiate them. So exo is again exit or outside. And so what an exotoxin is, it is a chemical product that's made by a bacterial cell. So a living bacterial cell makes this, but then it releases it outside of itself. And so the action is going to be outside of the cell. And mostly gram-negative have this, but there are some gram-positive that can make these exotoxins as well. And because these exotoxins can have different effects on different tissue types, we can also describe exotoxins based upon what cell do they cause damage to. So there are some exotoxins that are going to preferentially affect the nervous system of the host. So we can call those neurotoxins. Entero is a prefix related to the gastrointestinal tract. And so these are going to be really important toxins for bacteria that cause different food poisonings. So we call it gastroenteritis. But enterotoxins are a subtype of exotoxin. So again, those terms are really similar. So you have to spend a little time differentiating these. So exotoxin is answering the question, does a living bacteria make and release this toxin? That's what an exotoxin is. It has to leave and exit a bacterial cell. If that exotoxin preferentially binds to and disrupts the gastrointestinal tract, then we can also call it an enterotoxin. Okay. We can contrast exotoxins from endotoxins because these are not actively secreted. Endotoxin is a structural component of all gram-negative bacteria. More specifically, it is part of that outer membrane of a gram-negative cell wall. So remember, gram-negative bacteria have a specific cell wall. So if we do this kind of picture, here's a gram-positive, here's a gram-negative bacteria. One difference is how thick is the peptidoglycan. I'm not going to draw this whole thing, but thick. This one just has a few layers of peptidoglycan. Thin. So that's one difference. And then another difference is there is an additional membrane that only gram-negative have. So gram-positive do not have an outer membrane. And so that's where this molecule is. It's not actively secreted, but it's this molecule that exists in this membrane. So the time where it can be toxic is when gram-negative bacteria die. This molecule called endotoxin is released. So this, I just want to make sure it's clear as to what I'm describing here. This is not something the bacteria are doing on purpose, like a last minute, I'm just going to release this molecule because we're dying. All we're saying is when a cell dies, they don't remain intact. They start to degrade and just break into pieces. And so it's at the time of cellular death, and that can be your immune system is killing the bacteria, antibiotics are killing the bacteria, or the bacteria, inevitably cells just have a certain lifespan. And so when the cells, these gram-negative cells die, their parts are going to be released, and one of those parts that's released is this molecule called endotoxin. And it can lead to a variety of responses. And so this is what endotoxins tend to do to us. They have these general physiological effects where it can trigger fever, you can get aches, you can get pains, you can get tiredness, you can get shock where the different organs can start to fail. So it depends upon the microorganism and how much endotoxin is present. But that's one of the primary differences between exotoxin and endotoxins. So as we just mentioned, we have these two categories, exo versus endo. And there's a variety of differences that we can talk about. So as we mentioned, exotoxins have to exit the cell, right? So living bacteria release. So here's an example where a bacterium, again, has invaded our body. So again, this could be the respiratory tract, digestive tract, could be the skin, could be wherever it is. And this particular bacterium makes an exotoxin. So these living bacteria that are shown here as purple rods are making and releasing this exotoxin that can and does destroy these cells and tissues. So that would be an example of an exotoxin. And so it turns out exotoxins, you don't need a lot of them to be toxic. They tend to be highly specific, so there are some that can kill more types of cells, but some are often very targeted to specific types of cells. And they're usually these proteins that are unstable, so if you heat them up, they get destroyed. We can often take them and use them, as it says here, converted to a toxoid. And so this is important in vaccine generation. So the diphtheria vaccine, for example, uses a toxoid, so it's a modified toxin. It no longer has the toxic effects, but it's similar enough to it that it can stimulate an immune response. Release from live cells, and as we said, both gram-positive and gram-negative. Few gram-positive, but mostly gram-negative make these exotoxins. So that's the comparing and contrasting exotoxins versus endotoxins, but we still have to do this column. So endotoxins are again released by cell during the cell dying and lysing. So that's what's being shown here. In this case, we have a phagocytic cell. So that's an immune cell that's ingested and now digesting this bacteria. And one of the molecules that is now released is that endotoxin. And so this is dead gram-negative bacteria can release that endotoxin. So this is the first time in this lecture where we see this relationship of terms. So endotoxin, we can also call it lipid A, and it comes from this molecule that we learned about called lipopolysaccharide, or LPS. So those three terms there's some differences between them, but for our purposes, they're kind of, they're intertwined. And so endotoxin is part of that lipopolysaccharide in the cell wall, and it can be released when the cell dies. It's much less toxic, so you need higher doses of them. So bacterial endotoxins also, in addition to coming from the cell wall, or more specifically the outer membrane of gram-negative bacteria, it is more heat stable. And so because they're not proteins, they don't denature to the same extent as exotoxins do. And also because they're lipopolysaccharides or lipids, they don't lend themselves to developing toxoids, which can be used to stimulate immune response in the same way. The endotoxins tend to lead to a variety of signs and symptoms. So as we mentioned, it can trigger fever, it can trigger inflammatory responses, or diarrhea, or shock, or even coagulation occurring inside of blood vessels. So it is a variety of responses. And one other really important part of how endotoxins frequently work is it's a systemic response, and it leads to fever and inflammatory responses because it can trigger your own immune system in responding. So when we first introduced toxins, we were talking about
they cause damage to tissues, and so they can be direct damage. And so that's often how these exotoxins are going to be. The chemicals that the bacteria make directly target and damage specific tissues. Endotoxins, these molecules that are released during the death of a gram-negative cell, they often don't directly cause harm to tissues, but instead it causes our immune system to respond in a way that response leads to cellular and tissue damage and cellular and tissue changes that we perceive as that disease state.